Phosphorus. It's essential. What if it wasn't? In 2010, NASA-funded scientists claimed to have discovered an organism living in California that didn't need phosphorus. 15 years later, that paper's been retracted. What went wrong? G'day. Today I'm going to be discussing one of the most controversial papers ever published in the field of environmental microbiology, the Arsenic Life paper. I'm going to be covering the claims that were made, the evidence that was provided to support those claims, the lack of evidence in some cases, the backlash from the scientific community, and how we ended up today with that paper finally having been retracted. So join me for an episode of Tiny Giants. So, what's so special about phosphorus? Why is it essential? Well, one of the things that makes phosphorus so essential as an element is that it forms an integral part of DNA and RNA. Both DNA and RNA, or nucleic acids, are comprised of a sugar molecule attached to a phosphate backbone. Now, as the name suggests, the phosphate backbone contains phosphorus. The backbone of the DNA and RNA is necessary for these nucleic acids to form strands. It's the backbone that actually makes the strand on one side. The sugars then bind to complementary sugars on other strands to form, in the case of DNA, the double helix structure that we all know and love. And so phosphorus is essential because if you don't have phosphorus, then you can't make a backbone for your nucleic acids. If you can't make a backbone for your nucleic acids, then you can't make the nucleic acids. If you can't make nucleic acids, you don't have a genome and you can't express your genes. And if you can't do that, then you can't survive. Now, I mentioned arsenic in the introduction, and maybe you're thinking, why is arsenic relevant in a conversation about phosphorus? Now, the thing about arsenic and phosphorus is that they behave very similarly to one another in chemical reactions. If you look at a periodic table, you'll find that arsenic sits directly below phosphorus, meaning that the two elements have the same number of free electrons available to interact with other elements when forming molecules. Now, in turn, this means that the two elements effectively compete with one another to occupy the same space in a molecule. The reason that this is bad is because arsenic doesn't entirely behave the same way as phosphorus. And so if arsenic replaces phosphorus in a molecule, it can affect the way that that molecule behaves in downstream processes. Many of the processes in the cell that use phosphorus are essential, which means the uptake of arsenic into these compounds can be quite toxic. However, it is theoretically possible that an organism could evolve to replace phosphorus with arsenic in some of its compounds. And if an organism could do this, it would greatly expand the number of environments, not just on Earth, but in the universe, that life as we know it could occupy. The discovery then of an organism that's capable of replacing phosphorus with arsenic would be huge. And in 2010, that's exactly what a group of researchers funded by NASA claimed to have discovered living in a lake in California called Mono Lake. Mono Lake is interesting because it has a high salt content, a high pH, but most importantly, it has some of the highest naturally occurring levels of arsenic of any system that we know of on the planet. The team cultivated a bacterium from Mono Lake that they called Vreelandella GFAJ1, and which they asserted had the capability to replace phosphorus with arsenic in its nucleic acids. The key claim from the paper that it was capable of doing this was based on experiments that the group had done where they had grown the bacterium both with and without phosphorus. And what they found was that the bacterium could grow without phosphorus, but only if it was given arsenate to supplement. When they purified the nucleic acids from these bacterial cells, they then found that they got high amounts of arsenic coming along with them. They used this to assert that the bacterium had replaced the phosphorus that it usually used in its backbone with the arsenic. The paper that reported this was published in Science, which is the largest scientific journal based in the US. It was big news when it came out. NASA went so far as to hold a press conference to announce the results and to state that these results would change the search for extraterrestrial life as we know it. But things weren't quite as clear as they were made out to be. Almost immediately after this paper was published, criticism began to roll in. Now, over the years, there have been a number of issues raised with this paper, so I'm just going to focus on some of the more important ones. And none of them are really as important as the main issue, which is that the phosphorus-free media that the team had used to conduct their experiments on whether GFAJ1 could use arsenic instead of phosphorus wasn't really phosphorus-free. Some of the ingredients that the team had used had contamination, and that contamination meant that there was a final concentration of approximately 3 micromolar of phosphate in the media. 3 micromolar is not a lot, but it's enough to have an effect. In addition to this, the team hadn't provided a control experiment to compare the uptake rates of phosphate versus arsenate in their experiments. So there's no way to look at the data and assess whether or not the amount of arsenate being taken up makes sense for it to be replacing the phosphate in the DNA and RNA. On top of this, the experiments that the team performed never showed that the arsenate was actually in the DNA and RNA. All they showed was that if you grow GFAJ1 with arsenic and then purify DNA and RNA, you'll get arsenic with it. But it's entirely possible that this arsenic is just being purified alongside the nucleic acids and isn't actually part of them in any way, shape, or form. Following on from this, DNA with arsenic is estimated to be highly unstable. Regular DNA, which uses the phosphate backbone, is highly stable. It's one of the reasons that it makes a good medium to store our genomes. The half-life of regular DNA, which is the amount of time it takes for approximately 50% of the DNA to degrade over time, is around 30 million years. The half-life of DNA, if you replace the phosphorus with an arsenic, is around 0.06 seconds. That's so fast that even if the arsenic was in the DNA of Relandella GFAJ1, the techniques that the team had used to detect the arsenic wouldn't have been able to detect it quickly enough to see it. In the time since publication, the paper has received no less than 8 technical comments criticizing the methods or the interpretation of the group's original work. That's an insane amount of comments. Most papers never receive any technical comments. If a paper does receive a technical comment, usually it's 1, maybe 2. To have 8 is unheard of. Other labs have tried to replicate the results of the group and never shown any evidence of GFAJ1 taking up arsenic into its nucleic acids. Instead, what has been found is that when GFAJ1 is grown with arsenic, it selectively degrades non-essential molecules that contain phosphorus so that it can reallocate that phosphorus into more essential molecules like DNA and RNA. In response to the criticisms of their paper, the authors have always maintained that they believe that their interpretations were correct. They even went so far as to publish an FAQ responding to several of the criticisms of the manuscript in one location. And in 2012, when another group published results that showed that there is no arsenic in the DNA or the RNA of GFAJ1, they stated that they had new data that would be published within a few months. It's been 13 years since that statement, and no new data has turned up. The discovery of a life form capable of substituting arsenic for phosphorus would be a paradigm-shifting moment. It would greatly expand the number of environments, not just on Earth, but in the universe, that life as we know it could survive. Unfortunately, the data presented in the 2010 paper was just never sufficient to make the claims that were made. And data from other labs produced over the years has shown quite conclusively that GFAJ1 is not capable of replacing phosphorus with arsenic. Numerous calls over the years have been made for this paper to be retracted, given that the core claims that were made were demonstrably false. However, the paper falls into a bit of a grey zone when it comes to retractions. And the reason for that is that the authors didn't misrepresent their data. Not really. Their interpretations of the data certainly stepped way far beyond what was reasonable, given the results that they had. But that's a bit of a subjective viewpoint. They didn't manipulate the data or lie in any way. And given that, it's difficult for a journal to justify retracting an article if the article's authors don't agree to the retraction themselves. And as I said earlier, all of the original authors maintain that they interpreted their results correctly. In addition to this, there is actually a benefit to the journal at some level in keeping the paper in their journal. And the reason for this is that every technical comment not only cites the original manuscript, but it's also in the same journal. That boosts the journal metrics. Every published paper since then that references this paper also boosts their metrics. And so unless the reputation hit that the journal experiences as a result of having this paper in their journal counteracts the boost to journal metrics that they receive from all of the technical comments and the references, it's not really worth it for them in terms of a business point of view to retract the article. Now in this case, the weight of public criticism has been so much that in 2025, the paper was finally retracted. So there you have it. Despite any claims to the contrary, the search for arsenic-based life remains ongoing. Let me know down in the comments what you thought about this video, and hit the like button, subscribe, and notification bell if you want to stay tuned for my next video. And I'll see you in the next Tiny Giants.